distinguished speakers, Dr. Priyanka Gupta Banshal, Scientist D, ICMR, Headquarters, New Delhi. And the other distinguished speaker, Dr. Shagota Dasgupta, Professor, Department of Chemistry, IIT Kharagpur, West Bengal. Now, to chair this session, I would request Professor Nirmal Kumar Sarkar. Sir, please come up on the stage.
Dear participants, it is my proud privilege on this auspicious foundation day of this college to invite Dr. Priyanka Gupta Bansal, senior scientist from the headquarters of the Indian Council of Medical Research. I request Dr. Priyanka Gupta Bansal to kindly come to this podium and sit for a brief while when our students will be felicitating. Now, I take this opportunity to introduce Dr. Priyanka Gupta Bansal in brief to you all. Dr. Priyanka Gupta Bansal is an eminent researcher in the field of food and nutrition. Dr. Gupta Bansal has been working in Indian Council of Medical Research under the Ministry of Family Welfare, Government of India, since 2007 in different capacities. She is currently a senior scientist, though quite young by age, senior scientist in the Division of Nutrition at the headquarters of Indian Council of Medical Research. Dr. Bansal is a gold medalist from University of Delhi, and her core is in communities in India, maternal health problems, etc. Dr. Bansal has many publications in different national and international peer-reviewed journals to her credit. Dr. Bansal is actively involved in the capacity building activities of nutrition students throughout the country and has been organizing workshops in different parts of the country for promoting research, especially in home science and nutrition colleges. He facilitates and coordinates nutrition research and has been a part of several multi-centric studies carried out in the country on important areas of public health, such as micronutrient deficiencies, especially vitamin A deficiency, iodine deficiency, and amongst such problems amongst pregnant women. Today, Dr. Bansal has kindly consented to deliver a lecture on micronutrient deficiencies and ways to mitigate. So I cordially welcome on behalf of everybody in the hall, Dr. Bansal to kindly enrich us with her valuable deliberations.
Good morning, everyone. Am I audible at the last? Okay. Good morning and namaste. Thank you, sir, for your kind words. And to begin with, I like to thank the college of Midnapore City College to give me this opportunity to be present amongst you all to deliver little of my knowledge on micronutrient deficiencies. I hope you understand Hindi also, because I like to speak in dual manner so that I can get across and reach the audience. So through my presentation, I want the students to at least take some home messages about micronutrient deficiencies. So I would be speaking on micronutrient deficiencies and ways to mitigate the same. Next slide, please. Can I change the slide? So when Dr. Pradhan approached me about this conference, I asked her to tell me about the various technical sessions. I found out that one of the sessions is on biotechnological innovations to mitigate micronutrient deficiencies. So at that time I got an idea, why not to brief the students about the prevalent micronutrient deficiencies in our country and what are the usual ways to mitigate. Mitigate माने तो prevent कैसे करना है, control कैसे करना है, okay? So I would begin with what are micronutrients. As the word indicate, micro plus nutrients. मतलब कि nutrients जो कि कम amount में चाहिए होते हैं, small amounts में चाहिए होते हैं. However, though they are needed in the small amounts, that doesn't mean they are any less important. These micronutrients, which are primarily vitamins and minerals, are very, very important for optimum growth and development of the body. मतलब वो कम amount में चाहिए होते हैं, but at the same time वो बहुत ज़्यादा important हैं for our proper growth and development. So this slide shows the consequences. मतलब कि अगर micronutrient deficiency हमारी body में है, so what will be effect of those micronutrient deficiencies? As you can see, micronutrient inadequacies or deficiencies affect all age groups. Starting from infants, it can cause low birth weight, it can result in higher mortality rate and impaired mental development. Going on to children, it can result in stunting, against reduced mental capacity, frequent infections, etc. Adolescents, adults, pregnant women, elderly people, all are affected by these deficiencies. It's not only there is a particular age group that is affected. If we have these deficiencies throughout the lifespan, we will have consequences. It's not moving. Okay, so there are many micronutrient deficiencies which are there, but the prevalent in our country or the major micronutrient deficiencies in our country are iron, as you all know, which causes anemia. Then there is vitamin A deficiency, iodine deficiency, and vitamin D and zinc deficiency is also coming up in a big way. I'm not saying these are the only micronutrient deficiencies in the country, but here today I will be talking about just the major micronutrient deficiencies. So to begin with, let us see the trends of various deficiencies in the country. Trends मतलब कि prevalence. कितना deficiency है, उसका percentage कितना है. This slide shows the prevalence of anemia in preschool children. Preschool children क्या होते हैं? Less than 5 years. So if you see, this data is from NFHS, National Family Health Survey. The latest round that has come up for NFHS is NFHS 5, which is just out. So if you see the trend, you will realize that preschool children are suffering from anemia ever since the beginning. NFHS 2, which was done in 98-99, which is almost 20 years from now, you will see the prevalence has always been high. Be it 70%, 69%, 60%, that means 6 out of every 10 children in the country have anemia. This slide shows the prevalence of anemia amongst women. Women age kya hai? 15 to 49 years, because this is the age which NFHS collects data under. So here also you will see over the years from NFHS 2 to NFHS 5, the situation is still the same. And if you talk overall, it is more than 50% of the women in our country are anemic. Aadhi se zyada ladies, women are anemic in the country, and we have already seen the consequences of anemia. This is a very interesting slide. Now what this slide indicates is between NFHS 4 and NFHS 5. NFHS 4 कब हुआ था? 2015-16 में, and NFHS 5 कब हुआ? 2019-20 में. Now this slide shows the difference state-wise in the prevalence of anemia. You would not believe, out of the 22 states for whose the state report was out for NFHS 5, 16 states have shown an increase in the prevalence of anemia in the last 5 years. Our government is making so much effort. So ideally, the prevalence of anemia should decrease. It should come down. However, in the last 5 years, the prevalence of anemia state-wise, if you see, is also increasing. And in states like Assam and Mizoram, the prevalence has increased by 32%, 27% of the baseline value, which is a very alarming scenario. Now, this slide also shows the prevalence of anemia amongst pregnant women and NPNL women. What are NPNL women? Non-pregnant, non-lactating women. And here also the prevalence is nearly about 50%. So situation is not good in any age group. Moving on to Bitot spot, which is the manifestation of vitamin A deficiency. So Bitot spot is one area or one, I would say, intervention that has resulted in very good results. Bitot spot, which was around 1.8% in 1975, has come down to 0.2% in 2011-12. This data is not from NFHS because NFHS doesn't cover Bitot spot. This data is from NNMB, which is done by National Institute of Nutrition, Hyderabad. So here the prevalence has decreased, and it's a very good trend. Now moving on to this data is from CNNS survey. CNNS survey is Comprehensive National Nutrition Survey, which was conducted by Ministry of Health and Family Welfare in coordination with UNICEF. It was done in 2016 to 18. So this survey also shows that micronutrient deficiencies are quite prevalent in the country in all the age groups, 1 to 4 year children, 5 to 9 years children, and even in adolescents. However, if you see prevalence of vitamin D deficiency, which I've highlighted in this slide, it is very, very high. This is okay. Now, I have just shown you slides of some of the micronutrient deficiencies, but at the same time, the biggest challenge our country is facing as of today is dual burden of malnutrition. See, malnutrition is not only undernutrition. Malnutrition is nutrition which is not optimum. कुछ भी ऐसा जो कि normal नहीं है. तो जब हम बोलते हैं normal नहीं है, if you say whether my weight is optimum or not, तो अगर मैं बहुत पतली हूँ, मैं underweight हूँ, तब भी वो malnutrition है, और अगर मैं obese या overweight हूँ, वो भी malnutrition है. जब भी आप नॉर्मल से डिविएट होंगे इधर या उधर, दैट इज malnutrition. So what our country is facing today is double burden of malnutrition. When I say double burden of malnutrition, it means at the same time our country has underweight and at the same time it has overweight. So this is NFHS 5 data. This is NFHS data across NFHS 3, 4 and 5. You would see underweight amongst adults. This is data for adults. The underweight has decreased from 35% to 16%. However, overweight or obesity has increased from 9% to 22%. So you would see only fighting or addressing undernutrition is not our task. We have to address undernutrition, but at the same time also preventing the risk of overnutrition. So you nutrition guys, you have to look at it that your studies, your research questions should also address malnutrition, both aspects of malnutrition, undernutrition as well as overnutrition. Now, if I talk about strategies to control micronutrient deficiencies, कि अच्छा अब हमें ये सब पता चल गया कि we have micronutrient deficiencies and it is quite high in the country. But now what next? If I keep on saying we have micronutrient deficiencies, we have micronutrient, we all know we have. We have anemia, we have iodine deficiencies, we have vitamin D deficiencies. But what is more important than just admiring the problem is how to find a solution. So you all would agree there are three basic ways to mitigate or prevent micronutrient deficiencies, which includes supplementation, which is by and large the fastest way of correcting the deficiency. Then we have fortification, which is coming up in a big way. And then we have dietary diversity, which is the best of the three options actually. But it takes a very, very long time if you need to correct the micronutrient deficiencies just through the diet. So our government has been making a lot of efforts to correct these micronutrient deficiencies. There are so many national programs which have been started way back in 1960s and 70s to address these problems. So if you talk about the major or the main national programs for prevention of micronutrient deficiencies in India, the first one started in 1962, which was for iodine, and then for iron, that's correcting anemia, in 1970, and vitamin A program also started way back in 1970. So it's almost been more than 50 years. If you talk about IFA supplementation, which is iron and folic acid supplementation program, it was started in 1970, and at that time it was known as National Nutritional Anemia Prophylaxis Program. The program, these are, if a program is launched, after a few years, the government does the evaluation of the program, and if there is any change that is needed, so they revise the program. So this prophylaxis program was revised in 1991, and at that time it was called as National Nutritional Anemia Control Program. Then it was again modified in 2007, wherein adolescent girls were included. So what is the current program? When we take interviews and all for nutrition students for the various research posts we have in ICMR, this is one of the major questions, or we usually ask, what is the current program? But you know what happens, because the program keeps on revising over a period of time, sometimes students are not well aware of the recent program. So what I want to convey to the students here is that currently the program that we are following, the government program for anemia, it is Anemia Mukt Bharat, AMB. And this was launched in 2008. Now, what is the salient program, or the salient feature of this program? It is 6x6x6 strategy. What is this 6 into 6 into 6? 6 age groups, 6 interventions, and 6 institutional mechanisms. मतलब इस प्रोग्राम में 6 एज ग्रुप पे फोकस किया गया है, जैसे preschool children, adolescent, women of reproductive age, pregnant, lactating, like this. Then there are 6 interventions, जिसमें से एक है IFA supplementation, then there is deworming, then there is behavioral change communication, then there is fortification, etc. and 6 institutional mechanisms, which means there is inter-coordination between the ministries. ये सिर्फ health का program नहीं है, because anemia women and children को ज़्यादा effect करता है. So this is a coordinated program, जिसमें Ministry of Health भी involved है, Ministry of Women and Child Development भी involved है, public distribution भी involved है, like this. So these are the interventions which are being given under the AMB program. I won't go into the detail. Just to give you a brief, it is the provision of IFA tablets, then there is testing of anemia. They are also addressing non-nutritional causes of anemia. Then deworming and BCC. BCC is behavioral change communication, जिसमें आप लोगों को बताते हैं कि क्या अच्छा है, क्या गलत है, and you use visual or audio aids for it. जैसे flip charts, video films, etc. use करते हैं. Okay, now this is purely theoretical. I will not go into the detail, but I have already shared my presentation, and I would request the organizers to share the presentation with all the students, right? So this slide just shows you about the AMB program, what are the age groups which are being covered, and what is the supplementation they are giving, how frequently they are being given the deworming tablets and all. So this, you guys keep this handy, it will be useful. Okay, now moving on to vitamin A deficiencies. So what are the efforts that our government has made and what the program is doing currently? So this program was started in 1970, as I told, and इसमें करते क्या है, इसमें vitamin A supplement देते हैं, ठीक है, किसको देते हैं, pregnant women को देते हैं और बच्चों को देते हैं जिनमें vitamin A supplementation deficiency पाया जाता है. So this is the policy of government for vitamin A. What does it say? You know, it is a very old policy. It has not been revised. So maybe it's under revision and in a couple of years a new policy would come. But by the time that comes, this is what is existing. So they say regular consumption of dark green leafy vegetables and yellow fruits and vegetables because it's supposedly rich in vitamin A. Then breastfeeding, colostrum feeding is very important to prevent vitamin A deficiency. Then there is oral prophylactic dose which are given to infants and to children. So what is the dose that is being given? The first dose is 1 lakh IU international unit, which is given along with the measles dose at age of 9 months. And after that, from 1 year to 5 years, 6 monthly dose to children is being given for 2 lakh IU. तो अगर 1 साल से लेके 5 साल की उम्र तक 2 डोज हर साल देंगे तो कितनी डोज हो गई? 8 हो गई ना, और 1 डोज दी 9 मंथ्स पे, तो इस प्रोग्राम के अंडर टोटल कितनी डोज देनी होती है? 9 डोजेस. So a child who reaches 5 years of age should be given 9 doses. The next is iodization thing, which came up for preventing iodine deficiency disorders. This was started way back in 1956. ICMR did a study in Kangra Valley of Himachal Pradesh, which was very, very deficient in iodine. So they introduced iodized salt on a pilot testing basis. एक छोटे से district में iodine की deficiency को prevent करने के लिए iodized salt दिया गया. But the results were so successful and they were so encouraging कि ICMR की finding के basis पे government ने इसको एक national program बना दिया that we will sell only iodized salt. Okay, so here we have got a lot of success, and in India, large proportion of people are consuming iodized salt, and iodine deficiency is not that prevalent anymore. This is what I told you, that at present sale of non-iodized salt is prohibited, it is banned, and also the data is indicating, the national surveys etc. that are being done indicates that the consumption of iodized salt is pretty high in the country. It is very satisfying. Now in fact it's the other way around. Now people are working on hypothyroidism because it's being realized that many people are having thyroid disorders these days. So there is a thought that's going on that maybe this high prevalence of hypothyroidism is because of overconsumption of iodized salt. So that's also coming up in a big way. As I spoke and I told that diet is the main thing wherein, through which we should first try to prevent the micronutrient deficiencies. So you would all agree with me that we should eat healthy, including foods from all the food groups. Other than that, choosing all the food groups is one thing, but choose wisely is another thing. You are all nutrition students, you would agree with me that in iron metabolism in the body, inhibitors and enhancers play an important role. So we also have, as a nutrition students, in fact, we have to teach the community that, even through their normal diets, if they take care of inhibitors and enhancers of iron deficiency, they can prevent it to some extent. Inhibitors क्या होते हैं? हमारे फूड में कुछ ऐसे सब्सटेंसेस और इंग्रेडिएंट्स जो कि आयरन की एब्जॉर्प्शन को बॉडी में इनहिबिट करते हैं, उसको रोकते हैं, उसको कम कर देते हैं, ठीक है? तो उसके लिए हमें क्या करना होता है? कुछ मेथड्स होते हैं जिससे हम इनहिबिटर्स को रिमूव तो नहीं कर सकते, बट वी कैन रिड्यूस देयर अमाउंट्स, लाइक सोकिंग, मॉल्टिंग, फर्मेंटेशन, दीज आर सम ऑफ द टेक्निक्स. एंड एट द सेम टाइम इन्हांसर्स क्या होते हैं? Enhancers for iron absorption होते हैं. If we consume them in the diet, it enhances the absorption of vitamin C. It enhances its availability for the body. So you all know vitamin C is one of the enhancers, but the recent research is showing that some other substances and folic acid, peptides rich in amino acid, cysteine and vitamin A also may enhance the availability of iron in the body. But this research is ongoing. Other than your usual food that you are consuming, there are two other ways in which through diet we can improve and increase the micronutrient content. That is fortification and biofortification. What is fortification? Fortification is nothing but addition of some micronutrient to food, which is a superficial way. मतलब कि राइस बन गया, उसको आपने fortify कर दिया विटामिन ए से, आपने मिक्स कर दिया विटामिन ए को राइस में, जिससे कि उसकी availability and content बढ़ जाए. So fortification is a practice of deliberately मतलब कि जानबूझ के हमने उसमें डाला है, increasing the content of an essential micronutrient like vitamins and minerals in a food so as to improve the nutritional quality. And but fortified food कौन सा होता है? वो जो a large number of population is consuming. जैसे कि salt हर कोई consume करता है, किसी भी part of the country में रहता हो, या वेज हो या नॉन वेज हो. सो दैट इज वाई सॉल्ट इज वन ऑफ द व्हीकल्स विच इज यूज्ड फॉर fortification of iodine and now iron also. So what are the criteria for fortification? Nutrient deficiency should be widespread. जैसे कि selenium deficiency है जो कि बहुत कम है country में. So government won't do some fortification for selenium because it's not needed. They will go for fortification for just those deficiencies or those micronutrients which is quite prevalent. And the vehicle food should be consumed by the target group. The high consumption should not lead to toxicity. This is very, very important. कि जो आप मात्रा में fortification कर रहे हैं वो आपकी बॉडी के लिए हार्मफुल नहीं होना चाहिए. इट शुड नॉट क्रिएट टॉक्सिसिटी इन द बॉडी कि इतना ज़्यादा fortification कर दिया that there is an overdose in your body and it may lead to toxicity. So that is an important consideration, that the amount of fortifiant should be such that it should not lead to toxicity in the body. And it should not alter the taste, color, texture of the product. आपको राइस खाना है तो आपके राइस का एक आपका distinct taste है जो आपको पता है. If I give you some rice which tastes different or which looks different और उसका color different है, you guys will not consume it. So to make it acceptable, the color, texture and everything should be the same. This is the current situation of fortification in the country. That in 2016, FSSAI क्या होता है? Food Safety and Standards Authority of India. They operationalized the food safety and standards regulations for fortifying staples, mainly wheat flour and rice, and double fortified salt to reduce the high burden of micronutrient deficiency in the country. And जो fortified foods होते हैं उसमें +F का logo होता है so that everybody knows it's a fortified food that they are buying. And it was then notified by the Govt of India. And our Honorable Prime Minister made an announcement to mandate rice fortification in all social safety net schemes by 2024. And FSSAI is working on the proposal to make mandatory fortification for rice in the next 3 years. Now moving on to biofortification. Fortification क्या होता है कि food के अंदर हमने the food is ready and then you deliberately mix some micronutrient. But biofortification क्या होता है? जिसमें fortification is at the level of plant or crop itself. Okay. So a technique that aims to enhance plant crops with vital macro and micronutrients through biotechnology processes. And it is believed that it decreases the mortality and morbidity rates, which is associated with micronutrient malnutrition and gives rise to food security, efficiency, and quality of life for deprived populations. This is not my area. This comes under Indian Council of Agricultural Research. They have been working really, really hard, and they have come out with so many biofortified varieties which are rich in iron, zinc, etc. So these are some of the snapshots, like 17 biofortified food varieties which were produced by ICAR, and so these are different varieties which are rich specifically in one or the other micronutrient. So since this conference is for food informatics, so I just thought to also introduce very briefly what is the role of digital health technologies. You would agree food informatics में digital platforms होंगे तभी तो informatics develop होगा. So what is the role of digital health technologies in improving health outcomes? That is, the management and interpretation of big data could help to clarify the relationships and interrelationships of diet and disease as these relate to the socioeconomic and agricultural changes. I am not sure whether my audience is aware that there are so many nutrition informatics platforms which already exist in India. Okay, so one of them and the biggest of them is under Poshan Abhiyan. They have developed a platform under Poshan Abhiyan which they call as Common Application Software, wherein the data is pooled. And this gives you a ready source of information. Even if you go to Poshan Abhiyan site, you can go to their dashboard, you know, how many Anganwadis are covered, what the Anganwadis are doing, how they are tracking the child. And even in this common application software, if the pregnant, this Anganwadi workers is feeding the height and the weight of the child, then instantly she'll come to know whether it's stunting or wasting and where to go forward. And then, since this height weight is a regular thing, so next time when she will put in the height and the weight, then she'll also come to know the trend, etc. And it's all, you know, in the platform mode. So even the supervisors and the policy makers and the program managers all have access to this data. So rather than filling it in the register and all, and then making it digitized, this is already a digital platform that's available under the Poshan Abhiyan. And the same platform which was developed under the Poshan Abhiyan is also being used by under ICDS, that is Integrated Child Development Services. A dashboard is also made under Anemia Mukt Bharat. You all can go to their websites. There are very easy links that are available to these dashboards. Have a look. You will get to know you know, what kind of information is collected and how these digital platforms in India are helping us to, you know, actually know where our country is heading and what are the trends that are happening over a period of time. So this Anemia Mukt Bharat, it is compiling data on all the 6 interventions. They're compiling data and they're analyzing it to see the trends, etc. Then NIN, that is ICMR Institute, they have also developed a site for Nutrition Atlas, which provides information and data on the population's nutritional status. And then Nutrify India is a mobile app. You all can download it and see, you know, like the nutrients that you are eating, and you will get all the information on it. Niti Aayog has also created a state nutrition dashboard, which gives information on the health and nutrition outcomes on a state level basis. And Nutrition India is another platform that's made by MOH and FW. which also provides you information. So these are just lists to brief you and update you about these dashboards so that you can go and have a look. It's okay. This is just for the students. I have tried to compile a list of the major government policies and programs which are related to nutrition. So these are the lists. So when ma'am will share the presentation with all of you, you can have a look at, this is a consolidated list of the national programs and policies. So I would end my presentation stating that India is undergoing an enormous nutrition transition. Why I say nutrition transition? Because we are facing both undernutrition and overnutrition, and slowly our population is changing from undernutrition to overnutrition. So this transition is very worrisome. Double burden of malnutrition is the major nutrition challenge faced, and combination of the best approaches of fortification, biofortification, and dietary standards may be useful in combating micronutrient deficiencies in the country. With these, I like to thank you.
Thank you very much, madam, for your wonderful, compact, and very succinct lecture. Dr. Priyanka Gupta Bansal initially explained what micronutrients are. Then she explained the nature and consequences of different sorts of micronutrient deficiencies in the country. She also explained the status of micronutrient deficiencies, especially in children and women throughout the country. To add a bit humor, I could not understand why she disliked men from her arena of survey. Then she explained the dual burden of malnutrition, and it was surprising to know from her lecture that obesity is on the rise in the country. Finally, she explained the ways to mitigate micronutrient deficiencies with reference to supplementation and fortification programs involving iron, folate, vitamin E, and iodine deficiencies. The subtle beauties of her lecture were that I think she was the first speaker in this conference to highlight the importance of deworming, breastfeeding in mitigating MND, and who also highlighted the precautions to be taken during adopting different fortification programs. Thank you very much. And now the lecture is open for discussion. I would like to invite students to please ask questions of whatever is coming in their mind to Dr. Bansal. Students, please ask your queries. I think you can talk from there. Is it on? Okay. Put both of them on. Both.
Good morning, ma'am. I am Kea Das from VIH. Why in tribal area fat is deposited in the body than other nutrients?
I didn't get, can you repeat the question?
Why in tribal area fat is deposited in the body than other nutrients?
Fat fat is deposited in the body. You know, that depends on your intake. Anybody who will have higher fat intake, it will be deposited in the body.
Anyone else?
Another question is, there are any relationship between cardiovascular disease with micronutrient deficiency?
Yes, certainly. The recent researches are showing that these micronutrients also affects the pathways for the cardiac regulations, etc. So micronutrients like selenium, zinc, they do have a role in the progression and development of CVDs in the body.
Thank you, ma'am.
Good morning, ma'am.
Good morning.
Ma'am, my question is, iodine is good for the thyroid gland hormone synthesis, right? So why it cause hypothyroidism?
See, as you say, as we all know, excess of everything is bad. The problem that's happening now is, see, the government decided the iodization level in salt as 15 ppm at the consumer level and 30 ppm at the manufacturer level. So the companies have to pay a fine if the iodization level is below this. But unfortunately, there is no higher limit up to which they can iodize. Suppose I'm required to put 15 ppm, मुझे लगता है अगर वो कम हो गया तो मुझे फाइन देना पड़ेगा. तो मैं क्या करूँगी, 15 की जगह companies are putting 30 ppm, 40 ppm, because there is no regulation to see the higher amount of iodine. So that is resulting in a lot of iodine intake in our body, which is affecting in hypothyroidism. आपकी बॉडी को एक दिन में 150 माइक्रोग्राम आयोडीन की रिक्वायरमेंट होती है, 150 माइक्रोग्राम. वी assume that Indians consume 10 grams of salt per day. So considering 15 ppm in 1 kg, we say कि 150 microgram you will get. But now, because of the higher level of iodization which is being used by the salt manufacturers, you may be getting 250 microgram, 300 microgram. So we are leading towards iodine excess in the body.
Okay, ma'am. And the another question is, अगर हम मतलब कुछ फूड में अगर पहले से अगर कोई न्यूट्रिएंट प्रेजेंट है, उसके बाद अगर हम माइक्रोन्यूट्रिएंट जो है उसको अगर फोर्टिफाइड करते हैं, तो दोनों में क्या कुछ डिफरेंस है या फिर दोनों ही सेम वर्क करते हैं जब हमारे बॉडी में वो जाते हैं तो?
See, bioavailability is a major concern. Even from the if it is naturally present in the food, then the availability will certainly be higher, but that also depends on the inhibitors and enhancers in the diet. However, the fortified nutrient, its bioavailability is still a question. कि वो कितना आपकी बॉडी को मिलेगा. And it also depends on the physiological status of the person. Like for iron absorption, अगर आपकी डाइट में जितना भी आयरन है, in men only 3% iron is absorbed, whereas for women 5% is absorbed. That's how we are. We haven't done anything about it, but that's how our system is. So absorption varies according to physiological status, your age group, and also the diet that you are eating. आप खाने के साथ चाय पी रहे हैं या नहीं पी रहे, all these things would affect.
Thank you, ma'am.
Good morning, ma'am.
Good morning.
Welcome to MCC.
Thank you.
Can we call golden rice a biofortified food?
Yes.
Because you have told that the texture about the texture change and color change, because golden rice is too much yellowish than normal rice.
Yes,
So
yes.
that's why I.
See, golden rice was made way back when this fortification and biofortification was coming up. So what happens, over a period of time there is improvement. तो जो पहले product बना उसका color, texture and everything, it was not a very successful product. But now the varieties that we are being made and they are being consumed, they are much better because of the improvement in the technology.
Anyone else?
Ma'am,
Myself Shagota Dash. My question is that आपने मेंशन किया था अर्लियर अपने स्पीच में जो फोर्टिफिकेशन प्रोसेस है उसमें कुछ लिमिटेशन होता है, तो एज ए कॉमन मैन नाउ इन ए पीडीएस सिस्टम द fortified rice is involved, and how much the common man understand the fortification or fortified rice in this rice, or how much the limitation in this process, and how could she know or could he know that the fortified rice is present or not?
See, the rice which has been introduced in the PDS, it has already undergone trials for all the limitations that we discussed. जैसे कि एक limitation क्या थी? Content or the amount of fortification, right? So if that rice has been introduced in the PDS, it has already been taken care that it should not lead to toxicity. वो कैसे देखते हैं कि average consumption कितना होता है, male का भी और female का भी, 200 grams in a day or 300 grams in a day. So then we back calculate that this is the amount of vitamin A that we need in a day. This could be from other sources. So the vitamin A from fortified rice should not exceed this particular amount. They back calculate. So the rice which is introduced in PDS is very much safe, and it has been introduced taking into considerations all the limitations already.
Okay, thank you. But some ration providers are, we all know that which are not very efficient in his work or her work. तो इसलिए क्या कुछ अवेयरनेस कैंप चाहिए क्या जो हम कॉमन मैन को समझा सकते हैं क्या तो फोर्टिफिकेशन राइस?
Even if the distributors are like that, ये rice तो centrally procure and distribute होता है. If I want, I cannot increase or decrease the amount of fortification.
Okay, ma'am. Thank you.
Thank you, madam. It's really a nice presentation, and we are very much thankful that you have accepted our invitation and you came to our campus. We are really thankful
My
for
pleasure.
that. Actually, I want to ask you one question about that as you are very close to the ministry, so what is the thought of the government of India or the ICMR about the biofortification by the fermentation? You know, the some kind of the good bacteria, those are called the probiotic bacteria, they can fortify the rice, because in our surroundings we have a fermented beverage which is called a haria. This is basically a tribal drink. So we found that there are several kinds of the minerals and vitamins which are enriched after the fermentation. So are the government of India or the ICMR is thinking about that? This biofortification?
This doesn't, I'm sorry, but this doesn't come under the purview of ICMR.
Okay.
As I mentioned before, this is completely in ICMR's pocket, so we don't deal with agricultural aspect. We are a medical research organization, so we deal with once the product is there. If the government, suppose ICR has developed some fortified crop, then where ICMR comes in picture is conducting the efficacy and effectiveness trials for it. But what they are developing, what they are working on, that is out of, it's not in our purview.
So are the ICMR is funding about the biofortification by the fermentation?
No, we are not.
It's not. Okay. Thank you.
So thank you, madam, very much for your wonderful deliberations. And I hope our director, sir, Dr. Pradeep Ghosh and TIC, Dr. Kuntal Ghosh will be taking necessary initiatives to put your important lecture in the website of the college for wider circulation amongst the students. They will be Now it is my proud privilege once again on this auspicious foundation day of this college to invite an eminent professor from the Department of Chemistry, Indian Institute of Technology, Kharagpur, Professor Swagata Dasgupta, to present her lecture. I cordially invite Professor Dasgupta to this podium. At first, our students will felicitate Madam. Before I invite Madam to make her deliberations, it's imperative on my part to very briefly introduce this eminent personality to you all. Professor Swagata Dasgupta is associated with the Department of Chemistry at Indian Institute of Technology, Kharagpur. She has contributed to teaching and research at IIT Kharagpur for more than 25 years. Her research activities in biophysical chemistry revolve around proteins and encompass protein aggregation and protein ligand binding studies. Her research output is simply prolific. She has utilized research grants from diverse agents of Government of India and others. She has 160 plus publications in different national and international journals, peer-reviewed journals, to her credit. And she has produced over 30 PhD degree holders in her career. So she is prolific. She is a recipient of the Darshan Ranganathan Memorial Award from the Chemical Research Society of India. And she is also the recipient of the prestigious CRSI Bronze Medal a few years back. She is also a fellow of the Indian Academy of Sciences, Bengaluru. It's my proud privilege to invite Professor Dasgupta to make her valuable deliberations.
Thank you. Thank you very much for that very elaborate introduction. I'm delighted to be here. I come to this college very often, so I'm known by the faculty here, and I am thankful for this invitation to the organizers. What I will be talking about today is, as you can see from the topic, is since this is related to food, I also believe in natural polyphenols, natural diet, to the dietary constituents and how we can utilize them. But given my background in protein chemistry, what we try to look at is the interaction of these polyphenols with different types of proteins. Now, I will not talk of all the proteins that we actually work on, but two proteins that belong to what is called the ribonuclease superfamily. And for those of you who are comfortable or who know about what enzymes actually do, then if you look at the name ribonuclease, it tells you that this is a ribonucleic acid cleaving protein. So it will break up the RNA. So if we try to look at the specific aspects of what this protein actually does, I'll just walk you through the structure of the protein. So it's going to be a bit different when we look at what we mean by protein ligand interactions. So we have a macromolecule that is our protein, and we have a small molecule that is our ligand. Now this ligand interaction is going to give us information about where the protein binds. So where this small molecule is binding in the protein, it will give us that information. So giving us that information will tell us that if we want to design a molecule that is going to be a drug, that is going to inhibit the function of the enzyme, then I need something that is sitting in the same pocket. So if something is going to sit in the same pocket, I need to know the structure of this molecule. So that is what we mean by protein ligand binding. And this is absolutely at the molecular level. So I have a protein molecule and I have a small molecule. And I want to see how they interact and how I can modify the small molecule that is now going to behave like a compound or become a drug to inhibit the function of the protein. So if we go forward, So the question we ask is, why are we looking at polyphenols? Now polyphenols are potent antioxidants. They are specific nutrients that we have every day in our diet. So how can we utilize these specific nutrients? They act as antioxidants. They have anti-inflammatory, anti-carcinogenic properties, and they protect cells. So we want to utilize these. So how can we utilize these? So the next thing comes in the common dietary sources of polyphenols. All of us know that we have polyphenols in the fruit, in tea. So we look at specific molecules that are present in green tea. Curcumin, that is a very, very important polyphenol. So how can we utilize these in trying to understand how we can have them interact with proteins? And the then question comes as to what proteins. So when we look at the flavonoids, we look at their basic structure. Being a chemist, we look at the structural skeleton. This is called the structural skeleton. Now we have these different groups. These are called substituents. So as I change these substituents, I get different types of molecules. So I get quercetin, I get rutin, I get fisetin, a whole list of molecules that actually look like a whole slew, which I will show you later on. Many molecules. Now when I try and understand how these molecules are going to interact with the protein, I need to know how to approach the problem. So in the approach of the problem, I look at the structure. So I need to know the structure of the protein. If I do not know the structure, I do not know how the pocket is formed. So I need the structure. I need to understand the function, the binding, and all interactions of the protein. Only then can I understand what the specific interactions are, and only then can I design a drug or a molecule that is going to inhibit the function of my protein. So if I look at protein ligand binding in general, I need to have what is called steric complementarity. I need to have geometric complementarity and chemical complementarity. That is going to tell me there are different types of groups. Groups in the sense I have hydroxyl group, I have amine group, I have CH3 group. How are these going to interact? And what can I get from this interaction? So that is the question that we ask. The proteins of interest that we work in the laboratory, I have listed some of them here. Human serum albumin, ribonuclease A, angiogenin, lysozyme, and gamma-cystidine. Each of these we have looked at with our green tea polyphenols in understanding how they interact with these proteins. But I will today talk to you about only ribonuclease A and angiogenin. Now, to understand what these, so these are some of the work that we have done with human serum albumin when we looked at green tea polyphenols, curcumin derivatives, and other retinoic acid and different types of molecules to see how they interact with the most abundant protein in our body, the human serum albumin. Now, if we look at the ribonuclease superfamily, which is the study of the interactions that we looked at, why are these proteins so important? The proteins are very important because the first protein, pancreatic ribonuclease A, is the most well-known, most studied protein that has been used over ages to understand about protein folding, to understand how the sequence is related to the function, how the enzymatic activity is related to the protein. So when we try and understand or look at specific interactions with these proteins, we get an idea of how we can inhibit the enzymatic activity. So, of these different types, there are many different types of ribonuclease family. Now, they are called the superfamily because they all have a very common function, and that common function is cleaving of RNA because they are all ribonuclease type proteins. An interesting one of these is angiogenin. Angiogenin is a protein that is involved in a process called angiogenesis. Now, what angiogenesis is the building up of blood vessels. So, if I have a cut, then a tissue buildup will occur, blood vessels will form and heal the wound. But it is the same protein along with other growth factors that form a tumor. Now, every time I have a cut, I do not have a tumor. So, it is a very delicate balance of how we have angiogenesis inducers and angiogenesis inhibitors. But I have to have that blood vessel growth. I have to have that tissue growth to heal my wound. But I don't get a tumor every time. So to understand that, this is the process of angiogenesis. There is a bit of a color compatibility difference here because these are all red actually, showing you that there are blood vessels. So it all has blue blood now. You know what having blue blood means? Means you're all in royal company. So you have here angiogenic factors. Now what angiogenic factors do, they create through this blood vessel a blood supply. For any tumor buildup, there has to be a blood supply. So this blood supply creates a bigger and bigger tumor. Now what you want to do is you want to see whether you can actually design a molecule that is going to work on a protein and inhibit that. That would be an anti-tumor or an anti-cancer compound. So if we look at the protein, angiogenin, that is involved in this process, if we have excessive angiogenesis, we have several diseases. If we have insufficient angiogenesis, we have another set of diseases. So we want a very good balance. So this protein angiogenin is going to give us that balance. So to try and understand this protein, it is a small protein. So let's look at this factor. We have a tumor, we have a blood vessel buildup that creates a large tumor, and what you want is you want an anti-angiogenic factor. What is that anti-angiogenic factor going to do? It is going to diminish the tumor. So the question is, how do I do this? What do I look for? What do I need? And what do I target? So what we have done is we have targeted angiogenin, which is a protein that belongs to the ribonuclease family. So this is just an information about the protein. This is what the protein looks like. This part is the ribonuclease part that is involved in the enzymatic action. In addition, what it has a cell binding site because it binds to cells, and it has a nuclear translocation site. We're not bothered with that, but what we see here, it has an enzymatic site and a biological activity site. Now the interesting thing is these two functions are related. So if I can stop the enzymatic activity, it will stop the biological activity. So that is a good thing, or that is something that can be utilized as a chemist or as a biophysical chemist to try and understand how I can look at these interactions. Now, what we were interested in was a paper that came out in Nature that said that angiogenesis inhibited by drinking tea. So we were interested to look at the catechins present in green tea, and we found that from dietary constituents actually there is a lot of angiogenesis inhibition from curcumin, from pomegranate, from soya, from grapes, and from ginger. But we looked at green tea. But there is a whole lot of other dietary constituents that can actually be looked at in this specific form. So when we look at angiogenesis, inhibited angiogenesis, how do we do that? We can look at identification of specific dietary constituents. We can also look at synthesizing molecules. So those of us who know, I don't know any organic chemistry, so I have a collaboration. So we have an organic chemist, Professor Tanmay Pathak in our department, who makes the molecules, who designs molecules where we have a synthetic part. So we look at a dietary part, we look at a synthetic part, and there is a natural one. The natural one has been designed by nature. And if you look at the structure of this protein, it's just beautiful. This is the inhibitor, the one that is shown in blue here. So see how nature designs it. So I have a specific, so if this is my protein that I have to inhibit, nature has designed something that's shaped like this, so it just clops on the protein and it inhibits the function of the protein. So that is a natural inhibitor. So looking at how the natural inhibitor works is just amazing. This is the tightest protein-protein complex known to date because it just wants to stop the activity of the protein. So if we go to ribonuclease A to try and understand its structure, it has a specific, but what we are interested in is this one in blue. This is the enzymatic site. And I want to look at how I can, you have all studied enzyme kinetics, competitive inhibition, non-competitive inhibition. So if I want to look at competitive inhibition, I have to look at the substrate and try to mimic the substrate. So that is what has been done. So if we go further and try and look at angiogenin, it's very similar. And it has the same structure here. I think I have a better one on the next slide. So these are the two proteins. If you look at them, they're very, very similar. And the interesting part is here is the active site of the protein. Here is the, it's just rotated around. This is the active site. This is the active site of angiogenin. But in angiogenin, we have additionally a cell binding site, a nuclear translocation site, which is absent in ribonuclease A. So what I can do is I can target ribonuclease as a model protein for angiogenin because RNase A is widely available. I can do all my test studies on ribonuclease A, and then if it works with ribonuclease A, I then look at angiogenin. Then if I have an inhibition of the ribonucleolytic activity, I know that there is going to be an inhibition also of the biological activity in saying that it is anti-angiogenic. So, if you, those of you who look at structures, if you look at, they're very similar looking. So this is RNA. This is RNA, you know, the sugar phosphate backbone and the bases linked to it. I won't go into the details, but if you look at, this is the active site. This is for RNase A, this is for angiogenin. Very, very similar. Very similar because they all belong to the same family. So if I go a step further and I try to look at the design of now molecules that had been done, as I said, in collaboration, we look at specific molecules. These molecules are designed molecules that have been worked up in the laboratory that actually show inhibition. So showing that inhibition tells us that these molecules can be anti-angiogenic molecules. And that is what we are interested in. So if we look at ribonuclease inhibition, there is a whole set of molecules that have been designed, and we're getting better and better in the design of these molecules. So further on, if we look at our polyphenols. So which are the polyphenols that we are interested in? We are interested in the green tea polyphenols. These are the major polyphenols. These are the major polyphenols that we are interested in. And what we have here is EGCG, which is one that is looked at in a lot of papers. EC, epicatechin, epigallocatechin, EGCG and ECG, without going into the names. But this is the overall skeleton of the structures. So as a chemist, what we are interested in, I want to look at the protein and I want to look at the interactions. So when I look at these interactions, we have also done this with curcumin. All of us know about curcumin and how we can use these molecules to understand whether we have anti-cancer, antioxidant or anti-inflammatory activities. So these are some structures of anti-angiogenic polyphenols. So this is the whole list of polyphenols. A lot of them are anti-angiogenic in nature, but not all of them have been studied with angiogenin. So there is a lot of research left here. This has not been studied with angiogenin. We have done some with ribonuclease A, but not with angiogenin as of yet. So there is a lot of research here. So when we want to look at angiogenic compounds, what do we look for? We look at synthetic compounds, we look at natural compounds. And we try and see, when we look at these specific polyphenols related to green tea or curcumin, how we can inhibit the protein. So that is the question. So these are specific structures of proteins. You can, those of you who are interested in computers, can do just computer analysis to see whether we have the correct structure of the complex or whether we have the correct structure of the compound, and see how it is theoretically through computational chemistry sitting in the correct spot. So that is another aspect of what can be done here. We look at copper complexes. Now how do we do the experiment? We do the experiment by doing agarose gel. So what is, I won't go into the specific details, but just look at one of these. So this is agarose gel. So this is RNA. RNA is the substrate. If I have ribonuclease A and I add it to RNA, what is going to happen? It is going to chop it up. So I will not see the band, the intensity of the band you see is the least here. It means that I have RNA and RNase A. If I have RNA and RNase A has broken up this into small pieces, so I do not see it in under UV light. Now, if I have something that I have added mixed with the RNase A, and this is inhibiting the activity of the RNase A, it should be brighter, which means that there is less breakup. Less breakup means more of RNA, more intense band. So the fact that you can see a band intensity here means that RNA is there. If RNA is there, it means RNase A with whatever you have added cannot chop up the RNA, which means that is an inhibitor of the protein. So then we go a step further in trying to understand which is better. So looking at this, we go further. And so this is, so now if we just look at these band intensities. So this is RNA, this is with the protein, this is with the angiogenin rather. So same thing, because it has the same activity. So if it has the same activity, we'd have high intense band, less intense because now the protein has chopped it up. And now what do we have? We have more and more intense. More and more intense means better and better inhibitor. Because RNA is there, RNA has not been broken down. That means it is inhibiting the activity of the protein. So these are the specific ways we can look at these, where we have our angiogenin in this case. Now interestingly, if we want to try and understand the angiogenesis process, angiogenin breakup. Understanding angiogenin breakup means I have to have a live cell because I want to look at blood vessel formation. For this, what we have is we have a hatching egg. In a hatching egg, what happens? You have blood vessel formation. So this is where we have to do the experiment to see whether we have blood vessel formation. So the idea here is that we take hatching eggs, make a hole on the top. If you can see very carefully, there is a plastic disc here. So we have a plastic disc and we put our compound on the plastic disc, invert this over the membrane and then allow the chick to grow. So what is going to happen is if I add angiogenin, there will be growth. If I have inhibition, then there will be no growth. So how do I measure that? I measure that by the blood vessel thickness. So if I look at the blood vessel thickness here, here I have a control, normal blood vessel. When I have angiogenin, there is going to be a lot of protein because it is building up blood vessel. Now if I add the green tea components, you can distinctly see that there is thinning of blood vessel. Now if I add the angiogenin protein and the green tea component together, what is going to happen? It is going to look like the control. So very similar. So this is the test that tells us that we have an anti-angiogenic compound. This is called the CAM assay, chorioallantoic membrane assay. So this is the test for angiogenin. Now the question is now, if I just take the polyphenol, it is very dilute. So we have to devise methods that are going to give me a greater concentration. So to get a greater concentration, what we have done is we have prepared gold nanoparticles. So I have the gold nanoparticle and we've added the polyphenol to it. So now what happens? I have a large concentration of polyphenol. So now, because if I have just one single molecule of polyphenol, then it takes time for it to go to the target. So we have taken the gold nanoparticle and encompassed it with the polyphenol. So now if it goes to the target, what do I have? I have more molecules, so I can attack more proteins. Another thing that we have done is we have looked at curcumin, and beta-cyclodextrin is like a cup. So we have taken curcumin, it has taken this in the cup and it is taking it to the molecule. Because curcumin has a very bad bioavailability because of solubility issues. Okay, so beta-cyclodextrin is just a bunch of glucose molecules put together. So this is soluble, so it takes the curcumin and carries it along with it. So we have to have methods that are going to allow us. So we have the gold nanoparticle capped polyphenols, we have them with cyclodextrin encapsulated. So we're trying to increase the concentration of the polyphenols. So as we go further, so this is basically a picture where we have the gold nanoparticle and see how there are many molecules that can attach to it. So these are all the green tea polyphenols. Then what can I do? I can attach so many proteins to it. So in that case, I will be able to inhibit more and more proteins because that is the idea. I want to inhibit the protein. I want to inhibit the activity of the protein. So this is how we can look at the protein ligand interactions in a better way and then try and understand these. These are actually all red. You can look at the computer there, it's all red. Okay, but anyway, because these are all blood vessels. So then what we look at is we look at the specific type through the specific CAM assay that I just mentioned, and we try and look at the blood vessel thickness. And the blood vessel thickness, there are specific measures called the branching index, like if you have a blood vessel, like the branching of the arteries. Okay, this branching, this angle, this thickness, that gives you a quantitative measurement of how angiogenesis is proceeding. So we have methods, so look at this, this is all broken up. Okay, so what is happening here? It means we do not have blood vessel buildup. Okay, but the problem is that now if I have a wound, then blood vessel will not build up, then that is also bad. So I have to have a very delicate balance, as I said. So that is important. So we look at the different anti-angiogenic effects that we see here. So we have to see how we can utilize these anti-angiogenic effects. So when we look at specific relations or summary related to this specific topic, we do see that it does show anti-angiogenic properties. Now if we look at curcumin and how it interacts with the protein, we have similar ways in which we can look at the protein in trying to understand. So this is the structure of beta cyclodextrins. It's like a cup, and it takes in the curcumin and carries it along, because curcumin, as I said, has very low bioavailability, but it is a very good compound. Here again we look at the agarose gel. It tells us yes, there is inhibition. There is inhibition, and when you encapsulate it, you get better inhibition. So that is the idea behind this. We look at the specific kinetics, specific interaction probabilities of how one molecule is interacting with another with standard Jobs plots. These are typical biophysical studies that we have to do to understand protein-ligand interactions. Then we look at calorimetry, so we get the thermodynamics of the interaction that tell us about the entropic effect, the enthalpic effect, what kind of interactions do we have. Because that is the way you can design molecules. If you do not know the types of interactions you have, because as I said, we're looking at the problem from a molecular point of view. So we look at these specific assays. So again, when we look at the specific relation with the CAM assay for the curcumin and the curcumin beta CD, that is the beta cyclodextrin, we see that there is an increase in efficiency in curcumin because you have encapsulated it. So when we have the normal curcumin, we get inhibition, but when we encapsulate it, we get better inhibition. So we are trying to look at the polyphenols, methodologies by which we can increase the quantity of polyphenols that are going to attach to the proteins or going to inhibit the function of the proteins. Because in our dietary constituent, when we drink a cup of green tea, then we have these molecules as individuals. But if we can have them capped to nanoparticles from a therapeutic standpoint, then what is going to happen? Then I can deliver more polyphenol to my target site, which is what our idea is. So we look at the interaction of this and we find out, yes, we do have a better interaction when we put them on polyphenols and also when we add them to cyclodextrin. So another aspect that can be looked at is the theoretical aspect, which we also do in looking at this molecule and how I can, so I don't have to go to the lab, I can sit at the computer and just make the molecules and look and see how they interact with the protein from a theoretical point of view. Giving an idea, telling me that, okay, my molecule is sitting there and I will look at the specific interactions, then I say, okay, let me change this interaction. So I do it on the computer. And then if I see I get a better interaction, then you go for synthesizing the molecule in a wet lab or a synthetic lab. You don't go and make the molecules. You first think of, so you have to have a very logical approach. You say that, okay, this is sitting here, then how do I modify it so that it is going to interact better? So these are the specific types of interactions that you can look at from a theoretical standpoint. So when we're looking at these drug designs, so what we have is we have a ligand database. We can have, because you're at the computer, you can design how many molecules you want. You don't have to make them. You can design thousands of molecules. And you have your target protein, which is your angiogenin in this case, or my ribonuclease A, and I look for the specific interactions. Then I look to see how I can get a specific lead compound that can be developed as a drug for anti-angiogenesis. So that is the whole idea behind the research problem, or behind the whole thought process in targeting the ribonuclease family, in targeting ribonuclease A so that I can target angiogenin. we have a lot of work on this. So basically we are looking at the competitive and cooperative nature of the polyphenol capped gold nanoparticles and the curcumin beta cyclodextrin encapsulated particles. So basically what we are trying to do is understand protein ligand binding that is essential for the design and development of compounds that can eventually be drugs. So these are all my students, those who have completed their PhD, those who are still doing their PhD, the funding agencies, my collaborators, that without whose help I would not have been where I am today. And one quote which I believe in, which is very apt for this conference, this: let thy food be thy medicine and thy medicine be thy food. Thank you.
Thank you very much, madam, for your illuminating and high standard lecture. Professor Dasgupta initially talked about the basic principle of drug designing, and then she showed some slides on polyphenolic source foods containing polyphenols. Then she explained the pros and cons of polyphenol protein interactions, starting from the basics of protein ligand binding, choice of proteins, and why they chose ribonuclease superfamily of proteins. And she finally picked up angiogenin as the model for their studies. Also explained the process of angiogenesis, and then she covered how polyphenols obtained from green tea may act as natural angiogenin inhibitors. And then she highlighted the principles of assay of angiogenesis, or rather an assay of both angiogenesis and anti-angiogenesis. And she also covered the way curcumin or curcumin from turmeric can also be used for their studies. Now the lecture is open for discussion. At first, I would like to ask one thing: how does the addition of gold nanoparticles promote the evitation of the?
No, actually, what happens in the gold nanoparticles, since you have a larger surface, the molecules can sort of, yes, the polyphenols will sit on the surface, so you can carry more polyphenols to the target site. That is the idea, because you have a large number of protein molecules. So if I just carry one at a time, it is going to carrying a large number together.
One more thing: should people consume green tea more than ordinary tea, or
no, but
if I put the question other way around, does ordinary tea contain at all, or doesn't contain this polyphenols?
It does. Green tea definitely contains polyphenols. Green tea is good for you. Ordinary tea has thiaflavins. Those are also, but those are more, because you have a more processing going on, the polyphenols are a bit developed and they form thiaflavins. But what happens in green tea, you will see if I showed you the angiogenin, they thin the blood vessels. So everything is in limitation. It's not like you keep on consuming, because then you'll have too many polyphenols, which is again not good. No.
Thank you. Any question?
Good morning, myself Dr. Shantanu. I have a very silly question.
No question.
Questions actually is my personal queries. That in silico introduction, suppose gave this very good result, but have you ever faced that that best compound which is showing the very good introduction in silico model, it's failed or it's showing the less activity in wet lab?
Yes,
that is definitely. Can you share, if you share anything?
No, I haven't seen it per se, but that is definitely possible. Because see, when we're looking at a cell, Okay, there are cascade of activities going on. So as I said in the beginning, I'm looking at it from a molecular standpoint. So I have one molecule of protein and one molecule of ligand. But when I'm in the cell, there are so many proteins. It's like a soup in there. So you do not know what is exactly happening and how your protein or your ligand could interact with other proteins also. So that more studies are actually required. So these are called cell-based studies. So you have to do cell-based studies to actually see the efficacy of any compound. So the first thing that you do is you look at it from the computational point, you synthesize the protein, you do the basic experiments where you're looking at a single molecule. Then you go to see, first of all, before you go to any cell, you first of all see whether it's cytotoxic. So you do the standard MTT assay. So then you see whether it is killing the cells or not. So then if it is cytotoxic, then you go on to specific, say, cancer cell line studies. We've done that for some of other flavonoids, we've done it for some other flavonoids, and we've shown it for breast cancer cell line. We show that yes, it is working. But in those cases, we have just looked at the polyphenols. Say we have looked at fisetin, we have looked at naringenin. So these are the ones that we have looked at there, not green tea polyphenols.
Okay, thank you, ma'am.
Want to know actually, very nice your presentation. My one question is, which one is the best nanoparticle in contest to the metallic, or in case of the others form of the polyphenol? Like in your presentation, particularly when you use the gold nanoparticle, then it is purely in the metallic system, metallic nanoparticle or whatever may be. But in case of curcumin, whose results are the very fast, and what are the reason behind that? And if the how to gold are eliminated from the any body, if we consider as a drug or in other way in the therapeutic basis.
Yeah,
very simple, ma'am.
That is a very pertinent question related to therapeutics. Since we do not go into that, but yes, gold nanoparticles are used because of and the concentrations that you require here are very, very low. Okay, you go to less than nanomolar. So in effect it does not have, but there are studies that relate to how you have the excretion or how you have the loss of the gold nanoparticle, because once the gold nanoparticle has delivered its target, which is the problem with all nanoparticle delivered drug, because now you have nanoparticle encapsulated drugs. So the question is, what is happening when it is delivered? So what is happening to the polymer shell, or what is happening to the, so people are going to see where they get to very low levels of trace elements that will not affect it. But now people are making nanoparticles of the polyphenols themselves.
Okay.
So but the problem with that is if it aggregates, then there is a problem.
This is the
right.
most
So that is another issue. But people are making taking small organic molecules and then preparing nanoparticles of the molecules only. So once the molecules go in the body, the idea is that it will break up because of the enzymes in the body, and it will deliver so there will be no residual effect.
Very nice efficacy also. But in one thing, in our laboratory also we have developed the different types of the green nanoparticle regarding the gold, or even also the
silver nanoparticle also people use.
Even also the graphene, particularly for the carbon. This is also used in, this is the most important questions when we interact in the AIMS in Delhi. Then doctor advised that we are not accepted your
that is the problem. Silver is used because silver, when you have like burns, for burns, silver is used a lot for burns. So for burns, if you do, then silver is used.
Thank you, madam.
Madam, can't you use any chelating agent for removing this metallic particles?
We did that for copper related, where you can have EDTA or something that will interact. But the problem is you do not know the metabolic effect after that.
Afterwards.
Yes, that is the problem with all medication in this.
And did you study this angiogenesis only in this chick embryo model, or?
Only CAM model.
So
I do not know much of biology. That is as much as I know, and my students
a lot more has to be done.
Yeah, absolutely.
Any other question? Yes, please. Louder, please.
Myself, Surjakanto De. It's a good presentation, madam.
Thank you.
Congratulations. I have some queries and questions, madam. First of all, beta cyclodextrin is well known for the increasing of the solubility of the drug, right?
Yes, yes.
Yeah. You have no target specificity of the beta cyclodextrin in my knowledge. There is no target specificity. Okay, madam. If you working in the in vivo system, right, with a gold nanoparticle, however the size I don't know, you mention not mention here. So after, if you the administration route, basically it should be orally or intravenously, right?
As I said, I'm just looking at a molecular level. I have no idea how it should go, or how. The idea that we have is what I want to do is I want to deliver more polyphenols.
Oh, you are not delivered yet, madam?
No, no delivery.
Okay.
For me, I have, as I said, in the test tube, I have my protein and I have only, say, only polyphenol. Then I take in the test tube protein and gold nanoparticle loaded polyphenol. Then I compare the efficiency of these two. And then I see when I carry the gold nanoparticle, or when I have the polyphenol with the gold nanoparticle, then I have more efficiency. So that is the idea.
Okay. Okay, madam. Thank you, thank you.
Okay. But there is a lot of research that actually has to go into this, because you want target delivery. You're talking about nanoparticles. Then the question like was asked, now what is going to happen to the nanoparticle after that? You stripped it of its, all the polyphenols are gone. Then what happens to these trace metals? Okay, can I actually deliver it in that fashion? Then all the molecules that are tried to be used are protein related now. So if I have a polypeptide shell, then I have protease in the body that will break the peptide chain. So then it will be excreted. Okay, but that again is a problem, because as soon as I inject something with the polyphenol or with the polypeptide shell, it will break right away. It won't reach the target. So there was this very interesting study where what they did is, if you have like a tumor here, they prepared the nanoparticle that was tagged with iron. Okay, because iron is in our body. Okay, so they tagged this with iron. Then if this is the tumor, they inject the tumor here, inject the medicine here, that with the nanoparticle with the iron attached, and they put a very strong magnet. Then what happens is the medicine remains there. Because of the iron tag, the nanoparticle will remain there. Very simple idea. But that is the way you are ensuring. But again, too much iron will have a problem. So that balance is very, very crucial. So these, to go to medical, clinical trials is a long way ahead.
Thank you, madam.
Any other question from anyone else? So thank you, madam, very much for kindly consenting to deliver your lecture today on this auspicious foundation day of our college. Thank you once again. The scientific session continues just after 1 or 2 minutes.
Dear participants, please do not leave the hall. The session continues. We have given just 1 or 2 minutes break for having a cup of tea. Kundal, announce kore dichi. Research scholars who could not or did not present their papers yesterday should get ready now for presenting their papers. Research scholars who could not or did not present their papers yesterday should now get ready for presenting their papers. Kuntal. Research scholars who could not or did not present their papers yesterday, please raise your hands and be seated over here only so that I can see you. Research scholars who are willing to present their paper today, please be seated over here so that I can see you. Any one of you, please come over the podium. Who will be presenting first? Please come forward. Your name, please. Kon number dakho to? Number 5. Yes, please. You will be given a duration of only 8 minutes. You have to complete your lecture within 8 minutes. Start right from the objectives, aims and objectives, and skip this introductory part. Please stand over there. এই সাইডে না দাঁড়িয়ে ওই সাইড থেকে কর। It is Miss Pipika Das who is presenting her paper, Anti-Adipogenic Potentiality of Linoleic Acid Isolated from Tapra fish oil on 3T3L1 cells, a mechanistic study. Please start.
Good afternoon, everyone. I am Pipika Das, a research scholar of Biodiversity and Environmental Studies Research Center, Midnapore City College, under the supervision of Dr. Shravani Pradhan, Assistant Professor in Nutrition.
A bit louder.
Assistant Professor in Nutrition, Department of Paramedical and Allied Health Science, Midnapore City College. Today I am going to present my research work entitled Anti-Obesity Potentiality of Linoleic Acid Isolated from Tapra Fish, Ophistophthora staduri oil, a mechanistic study. Obesity.
Start from aims and
In the industrialized world, obesity has become the global epidemic associated with an increased risk of other life-threatening pathologies. According to WHO, over 1 billion people are worldwide are obese. Among them, 650 million are adults, 340 million are adolescents, and 39 billion are obese. These are the overall scenario of obese population in 2017. Obesity results from disequilibrium of energy intake and energy expenditure caused by the generation of adipocytes. Adipogenesis is the process of cell differentiation where pre-adipocytes mature into adipocytes. Several transcription factors are promote an adipogenic potentiality from the adipocyte differentiation involves in 4 stages, namely the growth arrest, mitotic clonal expansion, early differentiation, and terminal differentiation. Several transcriptor factors like PPAR gamma, CEBP alpha, SREBP1C are adopted as adipogenic regulators, and FAS, LPL, SREBP1P acts as a adipogenic effector. FDA has approved few drugs to treat or control obesity, but they have some adverse effect on human health. Recent studies suggest that fish oil, which is very low cost fish or marine fish, have the anti-obesity potentiality, so that we can choose tapra fish, which is cost effective and easily available in sea-coastal area of Bay of Bengal region. And at first we got, after GCMS study, we got linoleidic acid from tapra fish oil, and we check the anti-obesity efficacy of this potential compound. Therefore, the main aim of this study to investigate the anti-obesity potentiality of linoleidic acid isolated from tapra fish oil in vitro model or cell line model. To fulfill this aim, several objectives should be considered. First one is to investigate the cytotoxicity and anti-adipogenic potentiality of linoleidic acid on 33L1 cell line, and to search out the potentiality of linoleidic acid on anti-adipogenic or lipogenic markers on 33L1 cells. To fulfill those objectives, I have adopted several methods. First one, I culture 33L1 cells. At first, the cells were obtained from NCCS Pune and cultured to confluence at 37 degrees centigrade with a humified 5% CO2 atmosphere with DMEM high glucose media and 10% FBS and 1.5% antibiotic antimycotic solution. And after proper confluency, I treated linoleidic acid on different doses, that is 2.5, 5 and 7.5 micromolar per ml. And then I have checked cell viability assay for cytotoxicity and Oiledo staining for lipid accumulation and determination of lipid content. Then fluorescence microscopy by DAPI and anti-adipogenic or lipogenic gene expression by RNA isolation, PCR and cDNA synthesis, and then adipocytokine quantification by ELISA and Western blot analysis. First, we have studied the cytotoxicity assessment by MTT assay on cell viability of 33L1 cells. At first, cells were cultured on 96-well plate, and after proper confluency, I have treated linoleidic acid on 3 doses, 2.5, 5 and 7.5 micromolar per ml, and the figure 1 shows that there is no significant changes of cell viability after 24-hour treatment of linoleidic acid, indicating a non-cytotoxic effect of linoleidic acid on 33L1 cells. Then I have checked the lipid accumulation or determination of lipid content by Oiledo staining. At first, cells were cultured in 6-well plate and treated with linoleidic acid on different doses. First, cells were washed with cold PBH, and after that the cells were stained with filtered Oiledo stain solution, and the absorbance was measured 490 nanometer using a microplate reader. Figure 2A shows that compared to the untreated group or control cells without any treatment, the 2.5, 5 and 7.5 doses of linoleidic acid significantly reduce lipid accumulation decrease the number of lipid droplet, and 5 micromolar is the most effective dose of this treatment. And after staining, the cells were dissolved in isopropanol, and the absorbance was measured at 540 nanometer in using a microplate reader. And figure 2b shows that there was a significant decrease of lipid percentage after linoleic acid treatment, and here is also 5 micromolar dose of linoleic acid shows the strongest inhibitory effect of lipid accumulation. Then I have checked the fluorescence microscopy using Duppy staining. Duppy staining was used to assess the number of nuclei and as well as the gross cell morphology. Figure 3 shows that compared to the untreated group, linoleic acid significantly reduced the number of adipocyte nuclei as well as the number of adipocytes. Then I have checked the expression of adipogenic and lipogenic genes. At first, mRNA were isolated from a kit method, and cDNA and total RNA was quantified by nanodrop, and cDNA was synthesized by according to manufacturer instructions, and semi-quantitative PCR was conducted. Here, GAPDH is the invariant control. All mRNA expressions were normalized to GAPDH. Figure 4 shows that there was a significant increase of PPAR gamma, leptin, adiponectin and LPL. Those are directly inhibit adipogenesis, and SREBP1C and FAS was significantly reduced after the treatment of linoleic acid. Then I have checked immunoblotting analysis. Here all protein expression were normalized to beta-actin. PPAR gamma is the central regulator of adipogenesis or adipocyte differentiation, and leptin is also control food intake. It can stimulate satiety center of hypothalamus. After the treatment of linoleic acid, we showed that PPAR alpha, leptin and adiponectin was significantly reduced compared to the untreated cells, and the PPAR gamma, which increased the adipocyte differentiation, which are significantly reduced after the treatment of linoleic acid. Then I have checked the to delineate the specific molecular mechanism which I have studied. That's why I have checked the quantification of adipocytokine production or the secretion of adipocytokines in cells or media. Then I have checked the ELISA assessment. PPAR alpha, adiponectin and IL-1RA, which is the main anti-inflammatory. PPAR alpha, adiponectin and IL-1RA, which is an anti-inflammatory cytokines. Secretion was increased in after the treatment of linoleic acid compared to the untreated 33L1 cells. This is the overall work of my research work. At first, tapra fish was collected from Bay of Bengal, and fish oil is extracted, and then we have isolated linoleic acid and treated 33L1 pre-adipocytes, and I have checked the lipid accumulation as well as the number of cells by Duppy and adiponic and all the gene expressions which are directly related to lipogenesis and adipogenesis, and our results showed that there was a significant reduction of SREBP1C, PPAR gamma, which are directly related to adipocyte differentiation, and SREBP1C is the key regulator of adipocyte differentiation, and FAS is also involved in fatty acid metabolism or synthesizing palmitate or stearate or the saturated fatty acid, which are also reduced in our study. This result indicate that linoleic acid have strong anti-adipogenic potentiality of on 33L1 cells. This result showed that linoleic acid could be used as a lead compound to prevent obesity, but this is a dose-dependent study. Further research will be needed as a duration-dependent study, as well as in vivo experiment will be needed. These are some reference which I've taken, and
What may be the reason for the 5 microliter dose
5 micromolar dose is the most effective dose.
is more effective
Yes,
than a lower or a
higher dose.
What is the reason? What may be
Actually, it is a dose-dependent study. We don't know.
dose-dependent study, but the results were not dose-dependent. You found that 5 micromolar concentration is having the highest.
We check the different doses. What doses are most effective in this study?
Why just 5 micromolar concentration was
Because linoleic acid is a conjugated fatty acid, is a geometric isomer of linoleic acid. Omega-6 fatty acid also have various controversy of inflammation. That's why we check the anti-inflammatory or anti-obesity effect and which dose, which specific dose are related to adipogenesis or anti-adipogenic potentiality.
why that particular concentration is working better than a lower or higher.
Because omega-6 fatty acid sometimes inflammatory in higher doses. Thank you.
Next speaker, please. Start, please. Be precise from the very beginning. You must concise your introductory part.
Yes,
You
sir.
have to start right from aims and objectives part. Start.
Good morning, everyone. Now I am going to present a research paper on biocontrol of Vibrio herbae using a bacteriophage VP-MCC14, a representative of a new genus in the Curoviraceae class, under the guidance of Dr. Kuntal Ghosh, Assistant Professor, Teacher in charge of Medinipur City College, Department of Biological Science. India is the largest shrimp culturing country. Many people are economically dependent on this shrimp culture, but this shrimp industry are seriously affected due to some bacterial infection such as Vibrio herbae, Vibrio parahemolyticus, Vibrio natrigen, etc. Vibrio herbae is the main causative agent which is responsible for luminous vibriosis and the mass mortality of shrimp aquaculture. But the uses of antibiotics, there are to control this bacterial infection, shrimp farmers use some antibiotics such as tetracycline, chloramphenicol, ciprofloxacin, etc. But the uses of antibiotics are banned by Food and Drug Administration and the Marine Product Development Authorities. However, the uses of antibiotics have several problems. Vibrio herbae can resistance to these antibiotics and the chemical may accumulate shrimp tissue. This process called bioaccumulation, this causes rejection of shrimp product by major shrimp exporting countries. This terms the quality assessment failed. So there is need alternative of antibiotics. Bacteriophages have been suggested to be a substitute to the control of this bacterial infection in shrimp aquaculture. The main aim of this study, biocontrol of Vibrio herbae, which is a detrimental pathogen in shrimp aquaculture. The objectives are the isolation and characterization of Vibrio herbae infecting broad host range lytic bacteriophage, then the whole genome sequence analysis of suitable bacteriophage, then application of Vibrio herbae infecting bacteriophage for the controlling of Vibrio herbae in shrimp in laboratory condition. The methodologies of this study: collection of sample from different environmental sources, then isolation and propagation of bacteriophages, then host range analysis. After the host range analysis, the one-step growth assay, then liquid culture inhibition. After that, the stability testing. Then the genomic DNA isolation, whole genome sequence analysis of suitable bacteriophages, then the genome annotation, then comparative genomic analysis, then the application of Vibrio herbae infecting bacteriophage for the controlling of luminous vibriosis in shrimp culture in laboratory condition. First of all, we have isolated total 15 Vibrio species. Vibrio herbae were identified species-specific primer Vibrio herbae VHRF and VHRR, followed by 16S RNA sequencing. Then we have tested total 60 different type of environmental sample. Among of them, only 14 bacteriophages to be isolated. Among of them, one bacteriophage MCCCON. This bacteriophage, the host range, host is Vibrio herbae S5A. This is the plaque morphology of isolated bacteriophage MCCCON. The one-step growth assay of Vibriophage MCCCON, the latent period is 30 minutes and the burst size is 50. The liquid culture inhibition of isolated bacteriophage MOI1 took the 4 hour and the MOI 0.1 took the 5 hours. It confirmed that this bacteriophage could clearly lyse the host bacteria in aquaculture. After the stability testing of bacteriophage MCCCON, it could survive the wide range of pH 3 to 9, and after the stability testing of temperature, it could survive 4 to 45 degrees centigrade and the salinity up to 40 ppt, which suggests that this bacteriophage could survive in aquaculture environment. This is the circular genomic view of bacteriophage MCCCON. It was constructed by CGView server. Some putative functional protein are labeled. Bacteriophage MCCCON was deposited in gene bank NCBI accession number ON922990. This is the genomic and proteomic features of bacteriophage MCCCON and other ICTP classified Podoviridae, Stravoviridae family and Gorgonviridae subfamily. And the NCBI reported Myoviridae family bacteriophage. The bacteriophage MCCCON genome length is 134470 and the guanine and cytosine content for 34.5% and the number of protein 240. Bacteriophage MCCCON showed the similarity with the NCBI reported Vibrio infecting bacteriophage unclassified genus, highest similarity 18.54%, and the homologous protein is 127. Bacteriophage MCCCON was found the unique among of them reported Vibrio infecting bacteriophage, and it exhibited less than 18.54% blast and identity NCBI reported bacteriophage genome. Hence, we propose for the creation of a new genus in the Podoviridae class Vibrio infecting bacteriophages. This is the cluster sequence analysis of bacteriophage MCCCON. The bacteriophage MCCCON clustered with the NCBI reported unclassified genus Vibrio infecting bacteriophage. It was constructed by using cluster sequence analysis software package. The network was visualized 28262 after run. This is the dot plot of bacteriophage MCCCON. It was showed that the gray portion of the similarity with the NCBI reported unclassified genus bacteriophage. It was constructed by the GPART 1.40 program. The bacteriophage MCCCON genome compared with the NCBI reported blast and reported Vibrio infecting bacteriophages using the progressive move alignment. The color block indicates that 1-to-1 based alignment and the sequence similarity indicated by the similar plot within the NCBI reported bacteriophage. This is the phylogenetic analysis of bacteriophage VPMCC14. It branched with the NCBI reported unclassified genus. This is the phylogenetic analysis of bacteriophage VPMCC14. It was constructed by VIP tree, and it confirmed that the bacteriophage VPMCC14 clustered with the NCBI reported unclassified genus. This is the application of bacteriophage VPMCC14. It showed that after 15 days, bacteriophage VPMCC14 could control the Vibrio herbae S5 strain. This is the conclusion of my research paper. The bacteriophage VPMCC14 is significantly different in comparison of the other reported bacteriophage and might be designated a new genus in the Codoviracea class. Considering the survivability of this bacteriophage, extreme environment and the ability of the bacteriophage VPMCC14 lysis the Vibrio herbae S5a in stream culture, it can be confirmed that the isolated bacteriophage could be applied as a biocontrol agent in field trial condition. I would like to express my special thanks and gratitude our Honorable Director Sir, Medinipur City College, for providing necessary facility for research work. Dr. Kuntal Ghosh as a guide of my research work, then research scholar of Midnapore City College, IIT Kharagpur Swami Vivekananda Merit Cum Scholarships.
Stop there only. You were slightly faster than necessary. It was slightly more like reproduction from memory rather than slightly less than a lecture. You should talk less and let people understand what you want to say. You should be slightly more succinct and distinct. You said the phage can survive in extreme environments.
Yes, sir.
What do you mean by such extreme environments?
Aquaculture environment environmente 45 ডিগ্রি টেম্পারেচারে ওরা মানে বেঁচে থাকতে পারছে।
Did you
হ্যাঁ,
study it?
improved. Yes.
And lower level?
Lower
upper temperature?
Upper temperature 37 to 45 degree.
And
they can survive.
What is the low temperature?
They survive 4 degree.
But died at 0 degree?
0 degrees are normally decrease some numbers.
What was the range of temperature that you studied?
45 degree.
Up to 45, starting from?
4 degree to 45 degree.
You studied the effect of temperature only or any other parameter?
Salinity test.
Salinity?
Yes, sir.
What was the finding?
40 ppt up to survive this bacteriophage.
Up to 40 parts per thousand. And minimum level?
Up to 20 ppt.
Okay, thank you. Next speaker, please. You are Ananya Datta. Start presentation, your presentation.
Yes, sir.
And try to complete it within 8 minutes. Okay. So skip the unnecessary parts.
Okay.
Good afternoon, everyone. I am Ananya Datta, a research scholar of Midnapore City College under the Research Center of Biodiversity and Environmental Studies, and I am doing my work under the supervision of Dr. Shravani Pradhan. Today I am going to present my work entitled "In Anti-Cancer Efficacy of Tapra Fish Oil Derived Linoleic Acid on Human Breast Cancer Cell Line." At first come to the fish oil. Fish oil mainly derived from some fatty fish, and fish oil mainly contain omega-3 and omega-6 fatty acid, which is essential part of our body and which has many health beneficial effect, but these are can't produce in human body by itself. That's why it consumed by some supplementary food, and fish oil is the good supplement for omega-3 and omega-6 fatty acid, and it was statistically proved that day by day the cancer cases were increased in worldwide. That's why it need to treat it. So my aims of this study is to isolate fatty acid from tapra fish oil, which was used as an effective tool for cancer treatment or dietary therapy, and the objectives are highlighting the isolation and characterization of conjugated fatty acid as a single molecule to the develop anti-cancer drug from tapra fish, and to explore the anti-cancer activity of linoleic acid, a derivative fatty acid isolated from tapra fish. These are the methods which were done as per the standard method. This is the GCMS result after oil extraction from tapra fish. We were sent the sample for GCMS, and this is the GCMS result. And at the 20.37, these linoleic acid were present. There are many omega-3 and omega-6 fatty acid, but we were choose linoleic acid because it has anti-cancer potentiality, and before the linoleic acid were no data. After confluency of MCF7 means it culture in our lab, and after that linoleic acid were treated on MCF7, and after 24 hours at the 5 micromolar concentration, it result very effectively when compared to the untreated control group. And this figure shows that total nitric oxide assay and MDA level were increased after treated with linoleic acid compared to the untreated control group. And this is the parameter for reduced glutathione and oxidized glutathione. After treating with linoleic acid, the GSH level were decreased, means the apoptosis rate were increased, and GSHG level were increased, means apoptosis rate also increased. This is the gene expression for some inflammatory and anti-inflammatory gene, and this result shows that the inflammatory rate means TNF alpha is increased, because the TNF alpha is a gene that increase the ROS generation in mitochondria, and during the ROS generation the apoptosis rate were increased. That's why here the TNF alpha rate were increased, and the anti-inflammatory markers IL-1RA, IL-1R1, INOS and SOCS3 were increased after treating with linoleic acid compared to the untreated control group. And the IL-1 beta, which is a inflammatory marker, this is decreased after treating with linoleic acid compared to the untreated control group. And this is the Western blot analysis. Here P53 is a tumor suppressor gene, means when P53 is increased, the apoptosis rate also increase. And the IL-110, which is a anti-inflammatory marker, IL-1R1, which is also anti-inflammatory marker, these also increase after treating with linoleic acid compared to the untreated control group. And this is the ROS generation measurement by fluorescence microscopy, and this result shows that the ROS generation were increased compared to the untreated control group when treated with linoleic acid. This is the result for detection the apoptosis by using acridine orange and ETBR staining, and this result shows that the apoptosis rate were increased compared to the untreated control group when treated with linoleic acid. This is the result for detection of cell apoptosis by DAPI using fluorescence microscopy. DAPI mainly stain the nucleic acid, and when the cell were lived, then it bind with them, and here this result shows that the cell viability rate were decreased in when treated with linoleic acid compared to the untreated control group. And from this study, it was found that, or it may found that the which were the anti-inflammatory marker, these were increased, and which were the inflammatory marker, these were decreased. And so the which compound we were chosen means linoleic acid have anti-cancer potentiality. These are the canonical articles of my work. And lastly, I want to acknowledge our Honorable Director Sir, Dr. Pradeep Ghosh, Director of Midnapore City College, Dr. Sudipto Chakraborty, Principal of Midnapore City College, and my research guide, Dr. Shravani Pradhan, Assistant Professor in Nutrition, Department of Paramedical and Allied Health Sciences, and all lab mates of mine, and exam cells and administrative office, and Swami Vivekananda Merit Cum Scholarship for the financial support. These are the reference. Thank you.
You tried 2 doses, 2 microliter, micromolar or microliter, whatever it is, and 5. But another worker from the same laboratory,
Sir,
working
actually,
with the same acid,
actually,
tried
sir,
3 doses.
I have also the.
Let me complete my question.
Okay, okay, sir.
After trying only 2 doses, how could you come to the decision that the 5 microliter or micromolar dose will be the best choice?
No, sir, I have also 3 doses, means I done the work on 2 micromolar, 5 micromolar and 10 micromolar. After the 5 micromolar, 10 micromolar is more or less same to the control.
Then why did you not mention it in your slides? You are showing that you have tried only 2 doses.
Yes.
And you are rejecting one, accepting one another to be better. So I think you should reflect what you actually did. If you had used 3 doses or even more, you should have shown it in your slides.
Okay, sir.
Okay, thank you.
Thank you, sir.
Next speaker, please. Your name?
Good morning to all my respected faculty members, my fellow scholars, and the audience. So this is my presentation topic, understanding food security with respect to the soil organic carbon. Next, please. Next. So let me start with a question. And what do you think about that? When we think that what is the biggest challenge of an economic man? This is the fear of starvation. From the tiny microorganism to like big giant elephant, so all have a same common fear that how to collect food and for his and his family's survival. So this is as the biggest challenge of all the earning people. So from the early Neolithic age, it's like 10,000 years ago, we humans are developed and started agriculture, but still now not able to fulfill the need of all the humans and all times. So here's come the food security issue. So food security as a standard definitions given by FAO, the access by all people, all times to the food needed for a healthy and active life. The right to food should be implemented in the grassroots level in the all over the world. So the food security is the backbone of all the nations, and it is directly related to the well-being of the country. And worldwide, it's like 800 million people who are directly affected by the hungers. And in India, the number is 200 million. It's like 20 crore. And there is like 4 dimensions. This is like availability, access, utilization, and stability. And 5 categories of hungers are there: low, moderate, serious, alarming, and extremely alarming. And India falls into the category of alarming. So there is a lot of factors that affects the food security. So all these factors are centered around a single factor. This is increasing of populations, rapid increasing basically. So and there is also a lot of factors that infect the food insecurity. This is low income, and unemployment, poverty, and so on. And there is another aspect that is coming into the limelight now. So this is the trending issue. This is the climate change, and this is impact in food insecurity diversely. And then we have food waste. Talking about underdeveloped and developing countries, we don't have actually the proper infrastructure. Talking about the cold storage and packaging system. So this actually impact diversely and
talk directly on what you did.
Sir, actually I am starting this, So this is
information derived from internet. What
Yes.
you did actually.
Sir, I am starting on this actually.
Your time is limited.
Okay. So there is a lot of effect on the on our health issues and starvation, and that could leads to the permanent death. And there is no, look, I mean there is if children not get properly food, then it will he or she will not able to concentrate on the education field. And now look, previously yesterday when I starting this to make this PPT, I was searching that how much people are getting affected by food, and in the Google, then I was surprised that there is no such micro level data that could let me know that how much people are getting affected in Medinipur or Bankura or in this particular area. So this is, I think, the absence of micro level authentic data. And then there's the huge gap, and these gaps are basically can be fulfilled with the need of with the help of soil. So these soil are basically related to the soil organic carbon, and this soil organic carbon can improve the food production. And it's like soil organic carbon pool. If we increase the soil organic carbon pool by 1 ton carbon per hectare per year, that could increase the 30 to 50 metric ton. And this is the simple framework model. By looking this, we can easily say that the food security is directly related to the agri-business value chain, which is impacted by the management input, soil, and climate change. Climate, and this all the factors are related to the soil organic carbon, and this is direct or indirectly related to the food security. And there is a lot of management systems, so we can take some of the initiatives so that we can increase the soil organic carbon, at the same time food security also. So there is short and long term policy solution and diversification and so on. Thank you.
request all participants to kindly assemble in front of the Biological Science Building, where the Foundation Day celebration will be done within a very short time. Thereafter, you will have your lunch and then again come back to this hall for the valedictory session. So, please go to the in front of Biological Science Building. আচ্ছা ম্যাডাম, এখন তো বায়োলজিক্যাল সায়েন্স মেইন বিল্ডিং, in between these two main building and this building, on the ground, please assemble in the ground in between the main building and this building. Foundation Day will be celebrated over there. And after Foundation Day celebration, please go to canteen for having your lunch and then please return to this hall for the valedictory session.